Oh, it doesn't like that. Oh, wait, no. Okay.
All right. Welcome back to another Facebook Live. My name is Hannah. I'm coming to you from the Arizona Science Center. So for this week at the Arizona Science Center, our virtual camp is Calling All Scientists Camp. And in this camp, we're going to be exploring different STEM careers. And today, we're talking about careers in as medical technicians, as vet technicians. So this lab is called a urinalysis lab. And we're going to do a version of this lab that you can actually try out at home. Now, it does mean what it sounds like. So urinalysis refers to doing a lab looking at someone's pee or urine. And what are some reasons why we might do that? We might do that because we want to look for certain things in the urine that might indicate certain problems, like problems with type 2 diabetes, or that you may have liver disease, and we want to catch those things early so that doctors can treat it. So what we have here is since we're not testing actual urine, we have some different solutions here that have different concentrations of glucose and of protein. Those are two things that medical technicians often look for in someone's urine. So over here we have distilled water, we have water plus milk, we have electrolytes. So if you're going to the grocery store, that would be called Pedialyte in a lot of cases. So that has a lot of salts and sugars in it. And we have electrolyte plus milk here. So we have these strips that are called colorimetric strips. And they're called that because these felt pads change color based on whether there's certain substances present in a solution. So this first pad here, the one closest to the camera here, this is going to test for glucose, this darker green one. And this lighter green pad is going to test for the presence of proteins. We want to test for the presence of glucose in urine to look for signs of type 2 diabetes. Type 2 diabetes has to do with the presence of different amounts of glucose in the blood, and if you have too much glucose, that can cause or be an indicator for type 2 diabetes. And then with the second one, we're looking for proteins. Presence of proteins in urine can indicate a lot of different things, but that includes kidney disease and liver disease. So we're going to go ahead and start with our control over here. So this is our distilled water. Now it's pure water, and I call it the control in the experiment because we're not changing or adding anything to the solution. It's just pure distilled water. So when we test these different solutions, I'm going to stick this indicator strip just briefly in there. So really quick, just in and out. I'm going to place it on this paper towel, and it's going to take a couple minutes for us to see the results. So next I'm going to stick it in the water and the milk. All right. Next we're going to test our electrolyte or Pedialyte. So those are the kind of things you would also find in Gatorade or Powerade. Oops, put that over there. And last we're going to test our electrolyte plus milk. Okay. So we're going to leave those alone for a little bit, for a couple minutes. And in the meantime, I'm going to talk to you a little bit about pH. So these strips, these colorimetric strips that we're using to test for different things in urine, they're based on the idea of pH. Now, if we look at this bottle here, this is a bottle of pH strips, you can see that there are different numbers associated with different colors on here. Now, the pH scale is a scale from 1 to actually 14 that measures how acidic or how basic a substance is. So the warmer colors you see here, that represents more acidic solutions, and then the cooler colors here represent more bases. So when we think of acidic solutions, some familiar ones might be lemon juice or vinegar. And then if we think of bases, we can think of a lot of our household cleaners, such as ammonia and bleach. So we're going to do a very fun experiment called the milk of magnesia reaction to kind of demonstrate how pH works, all right? So we have this flask, is filled with water here, okay? And the first thing that we're going to add to it is called universal indicator solution, okay? So it appears green, and universal indicator solution is a chemical that basically turns different substances different colors to kind of tell us how acidic or how basic that substance is. So first we're going to add that in here and mix it around a little bit. Now we'll notice that this water is staying green. It's not changing colors because water is a neutral solution. So it would be kind of in the middle of that pH scale. The next thing that we're going to add is some vinegar. Now if we look back at our pH strips here, vinegar is an acid. So if we're going to make a prediction, we might predict one of the colors over here, which represents an acidic solution here. So let's see what color our mixture of vinegar and water and universal indicator changes to. We add our vinegar. We're going to mix it around a little bit. Now you'll notice that it's starting to turn red. And I might actually add a little bit more vinegar in there.
There it goes.
Yeah, just going to add a little bit just to make sure. Okay. So just like the colors on this container of pH strips, we're noticing that the solution turned red because we added that vinegar, that acid. Now the last step here is the most exciting. In here we have some milk of magnesia. Now if you haven't heard of that before, you can find it in the grocery store. You might be more familiar with Pepto-Bismol. It's basically a medicine that you can take for your stomach if you have a stomach ache. And it is a base. It basically acts to neutralize the acid in your stomach to make you feel better. So we're going to go ahead and add that, and we'll zoom in on here because this is going to happen fast. So notice that as I shake this around, it is changing colors. We had orange, and very quickly we have green, and now we have blue. So what's happening here is this milk of magnesia is acting to neutralize the acid of the vinegar, and that's why as I mixed around the solution, it slowly changed from warmer colors representing an acidic solution to more cool colors representing a basic solution. So what does this have to do with our urinalysis strips? Well, we're going to go back and check those out because it's been a couple minutes and they should be ready for us to take a look at. So these urinalysis strips, these felt pads, like I said, are colorimetric, or they change based on, or they change colors based on the solution and the presence or absence of certain things. This one is testing for glucose, this pad here, and this lighter green pad is testing for protein, all right? So they work in a very similar way to basic pH strips. There's chemicals that are basically part of this pad that, like the universal indicator, will change colors based on the pH of a solution. So let's go ahead and take a look at our distilled water first. So remember, distilled water is our control. So these are kind of the colors that we're going to be comparing the rest of our results to. And again, this is representing the glucose and this the protein. So glucose, our control is kind of a basic green color, and then for our protein, it's kind of a light greenish yellow color. So we're going to take this strip along with us and compare it as we go along. So first, let's look at our results of the water and the milk, all right? So for our glucose, I'm not noticing a huge difference in color, which is telling me, as the scientist, or as the lab technician, the medical technician, that I don't think that there's a huge presence of glucose in this water and milk. However, there is a differential in color when we look at the felt pad that's looking for protein. And it does look to me like there is protein in this mixture. And if you think about it, milk definitely has protein in it. So let's keep going here. Next, we're going to take a look at our electrolyte, or our Pedialyte. So with this one, the felt pad representing or looking for signs of protein looks the same color. So I'm not thinking there's a whole lot of protein in the solution. However, there's a big difference with our glucose. It's looking to me like there's a whole lot of glucose in our electrolytes. And that makes sense because glucose is indeed sugar, and sugar is one of the electrolytes that you would want in a drink to replenish yourself, especially after exercise. All right, let's look at this last one here. This is a mixture of both electrolytes and milk. So if we look at this strip, we see a change in color for both glucose and protein, which indicates that there is indeed glucose and protein in this mixture, which again makes sense because our electrolytes have that glucose inside, and then our milk has protein. So, again, this demonstration, we're not using actual urine, but it kind of shows you how you can use these strips to look for the presence or absence of certain things inside something like urine, and that can tell us if there's any signs in a person or in an animal that they might have a condition such as type 2 diabetes, liver disease, and other many important things. And if we are able to detect these things early, as lab technicians, we can address those things more early and help that person or that animal as soon as possible. So that's all I have for you guys today. Please join us tomorrow at 12:30 for our next Facebook Live. It's going to be all about aerospace engineering. And also, if you want more hands-on experiments or ideas in science and engineering activities, definitely check out azscience.org. Bye for now.